everybody. In this podcast, we're going to start to take a look at population genetics, and we're going to spend some time looking at some microevolutionary events and the factors that influence change in populations over what amounts to be shorter amounts of time. So let's start right in. Now, we know that there's variation. We saw this in the lab, and we know that we, even as a small group, have variations among the individuals that exist in our populations. And this is really important to the sustainability of the population over time. So there's this thing that talks about, one type of difference or having one difference structurally from one thing versus many differences structurally, and that's just what's going on between dimorphic and polymorphic. Qualitative versus quantitative, what are sort of the things we can see that sense to be differences, and what are things we can actually measure as being differences. So I can look and say, wow, it looks like there's differences because we don't have all the same hair color or whatever, but what can I actually quantitate and what can I measure to determine that variation? And within this, we're going to take a look at the concept of the gene pool. And all the gene pool refers to is the actual total of all the alleles that exist for those individuals in a population. So not what I have, I'm, myself, but what do all of those of my species have? What's the total amount of alleles that exist? Obviously, the more differences you have, the higher number of alleles you have, the bigger the gene pool. So the idea is that the frequency of the alleles in the population are going to change over time. Those changes will occur, and those are the changes and those shifts in the allele frequencies that are actually going to change the population and actually, therefore, then sort of springboard evolution. And when we talk about this, you're going to hear the term fitness used a lot, and we've used this term already, but fitness deals with how capable are you to survive, and also, not only that, here's something a little different, but how your offspring's survival is measured. So you can't just produce offspring, but your offspring have to survive too. So there's sort of this next level to really, really showcase how well will the population sustain. Okay. So as we look, let's take a peek at some of this variability. Well, DNA replicates extraordinarily truthfully since the beginning to now. We know that. We know that there's this high fidelity in DNA replication. So if that's the case, and mutations are very, 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 very small, where does the variability come into play? Okay, so with this is the idea that only mutations can actually create brand new genetic information. I can take a deck, I can reshuffle it. That's great. I might have a different order, I might have a new sort of situation, but reshuffling doesn't give me anything actually new. The only way I get anything actually new is if we actually add cards in, take cards out, and replace them with something else. So the only way that we actually get the new stuff is through mutation. Yet mutations are very, very, very rare, which is kind of interesting because one would think or hypothesize that the source of the variations that we have in the first place has to do with a mutation that occurred. So what could be some either latent or what we call the unseen variability causes? Well, recessive alleles, right? We can't see those. Somebody's heterozygous, so those are forms of it. The issue of multiple alleles comes into play, when you have alleles that, can only, we can only have two at a time. So, but if there are multiple alleles that exist, okay, those can be causes for change, okay? Polygenic inheritance, if you recall this, when we talked about the fact that you have multiple genes influencing a single trait. And then pleiotropy, which is in essence the opposite of polygenic. Instead of having many genes with one trait, which is polygenic right here, pleiotropy is one gene influencing multiple traits. And so these are ways that variability can exist without actually having anything new really in it. And then our big friend, epigenetics. So what kind of role is epigenetics playing? This is something we're going to see as time progresses and we learn a lot more about the effect those markers have on, you know, whether the methylation or the acetylation that occurs to our genes has on what can actually be passed on. So we know that with this variability, that natural selection in a particular environment is going to select or, you know, benefit a particular variation over another, and that's going to cause better chance of survival. So, but here's something that's neat. If you actually relax your selection pressure, the original, you sort of revert back to that original variation, okay? And this can happen with pesticides and resistance, you know, and that's why one of the new methods that they use, or that they even use with, for example, medicines to help keep viral loads down in people with HIV, is to actually rotate the medicines that they get, because that rotation constantly changes your internal environment, so which keeps the levels down and you're not selecting for a particular trait. So by rotating that, it never has a chance to revert back or actually select against one. So that natural selection is going to select against the phenotype, okay? And remembering that, because you can have multiple genotypes influence a particular phenotype. Think about dominance and the issue of being homozygous dominant and being heterozygous can have that same phenotype. So that's what it's actually selecting. So from this, we get into the idea of the Hardy-Weinberg Equilibrium. And Hardy-Weinberg is really interesting because this is a way that we can actually quantify or measure allele frequencies in a population over time. But there's a little catch to Hardy-Weinberg. There are certain conditions that have to be met in order to maintain the composition of the gene pool. So even with genetic recombination, we have to make sure we have that. So given certain conditions, the composition of a gene pool does not change, even if we have that, okay? So we can recombine, but no matter what, that frequency maintains the same, okay? So here are your conditions. You can't have any mutations exist. Try preventing that one. You can't have any migration, meaning there can be no gene flow. There can't be new individuals coming in from outside. There can't be anybody leaving, because that's going to influence the actual genetic makeup. You have to have a very large population. Mating must be completely random. And all the alleles must be equally viable. And that means there can be no natural selection. So a dominant allele, or for that matter, a recessive allele, one particular allele can't be, you can't be more adaptive than another. So let's look at how this works. So we have two very important equations we're going to look at. The P plus Q equals 1, where P is the frequency of the dominant allele. And I say that now because that's where we're going to start, but really just one allele. So, but we use P for the dominant. If you're in the case of codominance, you would just pick one allele to be P. Q would be the frequency of the recessive allele. If I only have two alleles that exist, the frequency of one plus the frequency of the other would certainly equal 1 or 100%. And but we're going to work in decimals, okay, for this. But in essence, those frequencies, we are talking about percentage makeup. The other equation that we're going to look at is P squared plus 2PQ plus Q squared equals 1. And this particular equation is going to allow us to determine particular frequencies of genotypes. Because if I were to look at a Punnett square, okay, and cross two heterozygote individuals, and instead of giving them the letters, I'm going to say, well, if they have one, then they have the other, and I actually cross them, well, that times that, I get P squared, that times that, I get a PQ, this and that, I get a PQ, and that, I get a Q squared, okay? So there's my P squared, so if I add up the total, I get that. Well, I have two of these, so there's my 2PQ, and I have a Q squared, so that equals that. So what this model allows us to do is to look at and see if over subsequent generations, is there any change that exists? So here's exactly what I just showed you, okay? We have the two heterozygotes, and you can see where those come from. This is going to be really important. Now I'm going to draw your attention to our practice problems. Okay, definitely spend some time going over these looking at them, spending some time with them. We will work pretty heavily in class and in the lab on these, so this is purely just an overview of the equations itself, just to give you that heads up. So work on your practice problems. You could even easily Google Hardy-Weinberg practice problems and come up with some. So have some fun, enjoy, and we'll be playing a lot with this in class. So take it easy, and we'll talk to you later. Bye.